I want to speak about our breeding work on Bambara groundnut, one of Africa's forgotten foods that has the potential to meet the demand for food and nutritional security in Africa. Our work aims to characterize a diverse set of germplasm for reproductive development under varied photoperiodic regimes, to develop segregating mapping populations for photoperiod and other agronomic traits, trait mapping, identification of linked QTLs, and marker development, to develop materials specifically adapted to different photoperiod requirements as a way to stabilize yield and improve the uptake of this underutilized crop. Our approach involves genetic crossing of Bambara groundnut in controlled environment rooms using divergent parental materials obtained from different African countries. This slide shows a cross between photoperiod less sensitive genotype IITA 686 obtained from Tanzania and highly photoperiod sensitive genotype ANKBA 4 obtained from ANKBA in Nigeria. After making genetic crosses, we use microsatellite markers to confirm the identity of these F1s and the segregating mapping populations before progressing with genotyping by sequencing to generate enough markers for genetic linkage mapping. This approach has helped in the development of a high-density genetic linkage map with over 1,200 markers covering the 11 chromosomes of Bambara groundnuts. And with the map, we have been able to identify a major QTL for pod number located on chromosome 4 in Bambara groundnut. Comparative mapping approach shows genome collinearity between Bambara groundnut and common bean for the QTL region, which means annotated genes in common bean will provide plausible candidate genes driving photoperiod response in Bambara groundnut. Bambara groundnut is a food security crop. It provides income for smallholder farmers in Africa. It can be used to develop products for consumers. It is important to diversify our food system that is overly dependent on too few crops. The crop is important as an animal feed. Although native to Africa, rich in protein, able to fix nitrogen, highly drought tolerant, and reasonably resistant to diseases, it is a short day crop with a unique photoperiod requirement. Till date, no improved varieties have been developed. Moving forward, it is important for us to breed varieties for key agronomic traits such as growth habit, podding habit, root architecture, seed number and size, tester color, removal of anti-nutritional factors, cooking or processing time, micronutrients, and resistance or tolerance to root parasitic weeds such as Electra. There is also the need for a better understanding of genotype by environment interaction that affects yield and how to select for genotypes that best fit agronomic practices and associated factors. Thank you for listening.